especially on the discrete section. They can throw in a question that is just out of left field, right? You got to be ready for everything.
Adam back for another MCAT podcast. We're done with passages for Biochem. Moving on to Discretes.
Thankful.
Last week's episode, I did well with the Discretes. So hopefully, we continue that trend. Highly doubtful, but I'm hopeful. I'm hopeful that we continue that trend. So, from a mindset, again, I love talking about mindset with all the different instructors from Blueprint Live Online. From a mindset standpoint, you finish a passage. Are you taking that 3-second kumbaya break? What are you doing knowing that your next path is into Discretes?
Yeah, so sort of the difference between going from either passage to passage or discrete to passage is that you don't have to worry about moving like efficiently through a pretty dense passage. So with discretes, I still do take my kumbaya, but it's not as much prepare for what is coming as much as it is don't worry about what just happened. Like maybe the passage didn't go great. So I'm still taking my kumbaya, but it's less preparatory and more, sort of keeping my mindset steady and making sure that I'm not worried about like anything that went wrong so far.
Nice. All right. So let's go ahead and jump in to our discretes here. I'll start for fun with question 57. Which of the following do not have proteins with a nuclear localization signal? And immediately I kind of sigh and I'm sad because it's a Roman numeral question. Roman numeral 1, E. coli. Roman numeral 2, Homo sapiens. Roman numeral 3, fungi. And Roman numeral 4, archaea. No idea what that is, but so which of the following do not have proteins with a nuclear localization signal? So what's up with Homo sapiens here? Obviously, answer choice 2, if you're watching this, is not in any of the answer choices. So apparently they do have proteins with nuclear localization rates. Screw you, test writer, for like throwing in this random thing. So E. coli, answer choice A. One of the strategies I've learned over the period, and let me know if you kind of agree with this, is just for Roman numerals, which answer choice shows up most commonly? Start there. And E. coli, answer choice 1, is the most common.
Yep.
So if it does not have proteins with a nuclear localization signal, then it has to be answer choice B. But if it does, then we have to go through the rest.
Well, actually, the important thing there is that since this is a not question, if it does have proteins with nuclear localization, that's what we would be able to say is answer option B, because this is sort of like a negation question here. Like you're saying, so it's only answer option or Roman numeral 1 is only going to show up if it does not have proteins with that signal.
Yes. I think I understand what you're saying. I thought that's what I said. So if E. coli does not have proteins with nuclear localization signals, we have to not. I'm super confused about what we're saying now.
If we decide that E. coli does not have proteins with this signal,
yeah, then
it could still be A, C, or D.
Correct.
Yes.
Yes. Okay. We're saying the same thing differently.
Yes.
Potentially.
Okay. Sorry.
All right. So the first problem is I have no idea what nuclear localization signals are. And the second thing is, well, protein. So what has proteins? Does E. coli have proteins? Or is it just this bacteria that doesn't have proteins inside of it? Obviously, my guess is that it would have to have proteins to kind of be made there. Maybe, but I don't know. So that's where I'm lost. I'm lost at the very beginning because, number one, I have no idea what nuclear localization signals are. And whether or not E. coli has proteins or not. That's just like, that seems silly. But cells have to have proteins in them to kind of be there. So that's neither here nor there. So how would you answer this?
So I'm looking at this. It's definitely important to know what a nuclear localization signal is. But if you're coming at this question and you don't, maybe you start to use context clues. Like you said, so none of these answer options have Roman numeral 2 in them. And we know what Homo sapiens are, hopefully. And so knowing what Homo sapiens are, we know that Homo sapiens do have a nuclear localization signal. So that's sort of just a little piece of like detective work that we can start with to sort of set us on the right track. So maybe trying to distinguish between Homo sapiens and any of these other Roman numerals, like what are the categories that Homo sapiens fall in? And, how does that differ from, and maybe any of these other types of organisms?
Yeah. So obviously fungus is a little bit different than a Homo sapien. E. coli is bacteria, kind of is with us and lives with us and does bad things to us. I have no idea what the Roman numeral 4 is. Archaea? I don't know what that is.
Archaea. I think that's a pronunciation.
Yeah, but it looks fancy. So I don't know if that helps. That fungus is like different enough to just say it's B and move on. So
if you have no idea, good, decent strategy is just like trying to find something different and like heading with it. So in this case, nuclear localization signal, this is like a hint that whichever, if you don't have proteins with a nuclear localization signal, then you don't have a nucleus. So we're looking for which Roman numerals or which type of organisms are not going to have a nucleus. And so then the distinction is if you have this outside information, prokaryotes are not going to have nuclei, whereas eukaryotes will. So really this question is which of the following are prokaryotes, because it's asking us which of the following don't have a nucleus. And so from there, this is just recognition. We're saying which ones are prokaryotes, aka which don't have a nuclei. E. coli, I know, is a bacteria, so that's a prokaryote. Okay, so 1 has to be in my answer. Homo sapiens, that's a eukaryote. Okay, I shouldn't see 2. Fungi, that's, even though it is quite a bit different from Homo sapiens, they are also eukaryotes, so fungi are also going to have nuclei. So we shouldn't see answer option 3 in there. And then Archaea, also a prokaryote, and so not having a nucleus. So we should see 4. And so we look into our answer options. We see only C has 1 and 4 only, and so we know it's C.
And I should have gone to Old Faithful, when in doubt, choose C and move on. And I would have been right, darn it.
Definitely.
So often. It's funny. My brain just being so far removed from this, and it's good to remember this. My brain almost never goes back to prokaryote, eukaryote in these questions. And it comes up so much that it's such a, like, if a student just took a split second to go, is there a prokaryote, eukaryote difference here? And if there is, then, that's probably, I'm going down the right path. And I just always forget to go there.
Yeah.
So.
Yeah, it can be so foreign because, especially if you're so removed from sort of just this like nitty gritty content stuff, remembering how we, divide organisms at like sort of like a basic level, like prokaryote, eukaryote type stuff. It can be hard to remember to go down that path. But so I definitely sympathize with you there.
Yeah. All right. Go ahead and read 58 for us here.
All right. While the blood is buffered primarily through the equilibrium between carbon dioxide and carbonic acid, coupled with hemoglobin, the blood may also be buffered through other plasma proteins. Which of the following is true? A, A shift in the pH can alter the tertiary and quaternary structure of the protein, allowing it to buffer the pH by precipitating out of plasma in response to pH shifts. B, the amino acid residues that make up the protein may act as Bronsted acids or bases, reducing shifts in pH. C, plasma-soluble proteins have enzymatic function, allowing them to sequester hydronium ions from the blood inside membrane-bound organelles in the podocytes lining the capillaries. Lot of terms in that one. D, in the presence of altered pH, any plasma-soluble proteins will undergo either acid or base catalyzed cleavage, thus depleting the acid or base, causing the disruption to blood pH.
Wow. So answer choice C just seems overly complicated. Like you're just trying to get too fancy with me. You're trying to confuse me. So just for that reason alone, I'm gonna hope that that's just a question writer trick. Right, the podocytes getting super specific. It's only in the podocytes that this is happening. I'm like, oh, okay, I don't know. Bronsted acids, I don't remember what those are. Answer choice A, a shift in the pH can alter tertiary quaternary structures of the proteins. Like, is that true? Like, I don't know. The structures of a protein seems like it's really important. If you're altering the structures of the proteins, like that seems like it's pretty significant, which seems like it's a little too far, but I could be thinking about that wrong. I like answer choice D because like we have all of these proteins and amino acids and which make up the proteins, and we can, are they basic, are they acidic, and like it sounds like it's like, yeah, that's what it's for, let's do it. So I like D just for that.
All right, answer option D. All right, so this is a pretty hefty question here. There are like a lot of terms. I like the fact that you eliminated C. Definitely this is like super specific, and not only is it super specific, it's actually not true. It's factually false. Like this is not what occurs. But let's go through these answer options one by one now. We've eliminated C based on the fact that it's overly complicated, as well as the fact that it's just false. But a shift in the pH, looking at answer option A, a shift in the pH can alter the tertiary or quaternary structure of the protein. Okay, that sounds right to me so far. Allowing it to buffer the pH by precipitating out of plasma. Okay, so this part seems wrong to me. So while changes in pH can denature proteins, the reason A is wrong is because it references precipitating out of the plasma. That's not true. That's not what happens with alters in pH. Although pH can denature protein and change the tertiary and quaternary structure. So this first component is correct. The second component about precipitating out of the plasma.
Yeah. I don't want precipitates in my blood vessels.
No, you don't. That's true. That's very true.
That seems weird. Yeah.
All right. Answer option B. The amino acid residues that make up the protein may act as Bronsted acids or bases, reducing shifts in pH. Okay, so this seems definitely reasonable to me because we know that amino acid residues, there are both basic and acidic side chain groups, aspartic acid or like arginine, histidine, things like that. So we know that side chains could act as Bronsted acids or bases. So this seems pretty reasonable to me. Gonna stick that one in my back pocket though. We eliminated C for its overcomplication and the fact that it's just not true. And then answer option D, in the presence of altered pH, any plasma-soluble proteins will undergo either acid or base catalyzed cleavage, thus depleting the acid or base, causing the disruption to blood pH. So D, what was your rationale for picking D again?
I don't know. It just seemed like it worked. I'm like, proteins, yeah, they have amino acids and they can do that kind of stuff.
I see. Yeah, so D is actually also similarly to C. It's not as term intensive or like super specific or wordy like C is, but D is also just factually incorrect. Like this is not a physiological phenomenon that we see. If plasma soluble proteins would undergo acid or base catalyzed cleavage, like in the presence of altered pH, our bodies would be in a hefty amount of trouble pretty frequently, because we have to, we have to be able to moderate those changes in pH. And if any change in pH caused a cleavage of our soluble proteins, yeah, we wouldn't.
I suppose that was a little extreme. Okay.
But okay, so I'm returning to answer option B because it seemed good to me, and I didn't notice anything that was explicitly incorrect in there. So the amino acid residues that make up the protein can act as acids or bases, reducing shifts in pH. Okay, yeah. So after, I wasn't like entirely sold on it at first. Like, even though it was true, I sort of wanted to explore my options with C and D, but definitely comfortable with B for the fact that I know that side chains can act as an acid or a base.
Alrighty. Answer choice B. All right, 59. Which of the following correctly lists a pair of analogous structures and a pair of homologous structures? A, the wing of a bee and the wing of a bird. The wing of a bird and the leg of a bird. All right. So this is like reading poetry here. So analogous structures, homologous structures. So analogous to me is like similar in terms of function. Seems like that's what analogous structures potentially means to me. Homologous structures, again, homo meaning same, but that would be like wing and wing, but analogous seems like it's more appropriate there. And so homologous to me potentially is something, I don't know, a little different. Anyway, answer choice B, the wing of a bee and the wing of a bird, the arm of a human and the flipper of a walrus. Okay. So that seems like homologous. Oh, it's an arm and an arm, a wing and a wing, though. Those are the same. I don't know. All right. C, the arm of a human and the wing of a bat. Oh, okay. And then the wing of a bird and the wing of a bat. Okay. Maybe I like that. D, the wing of a bird and the wing of a bat. Or the wing of a bee and the wing of a bat. Wow. All right. So let's just try to break this down. Wings help things fly. So wing of a bee, wing of a bird, wing of a bee, wing of a bird for bee. And then D, wing of a bird, wing of a bat. Like, that doesn't seem to be different enough for me. And then wing of a bee, wing of a bat. Those doesn't seem to be different enough for me to, like, make me guess analogous versus homologous. What's the difference between a wing of a bird and a wing of a bat versus a wing of a bee and a wing of a bat? Well, one is a mammal and one is a insect, but that's still wings, so they still help things fly. Although the wing of a bat is more of an arm. Oh man, this is really hard. I like C just because the wing of a bat similar to an arm with a thing, and then wing and wing of a bird and a bat. I like C just for that. But this seemed like it's making a fool of myself much more than it needs to be.
Yeah. So this question is very much so like, just, do you know what an analogous structure and do you know what a homologous structure is? You were pretty spot on with your sort of like analysis or sort of your logic to try to get to what an analogous structure was. So analogous structures are, best way to phrase it, structures, they evolved independently, but they have similar functions. So just like you recognize like the wing of a bee and the wing of a bird, they evolved independently. They have different evolutionary histories, but those wings serve the same purpose, flight. So for that reason, a lot of these in here are analogous structures. So the wing of a bee and the wing of a bird, that's analogous. And then that last answer option, the wing of a bird and the wing of a bat, that's also analogous. So D is wrong because both of those options are analogous. Yep. Just like you recognize, like they were too similar to sort of be like a comparison.
Yep.
So I like that you threw out D just sort of based on your reasoning between the answers, and that these seem too similar to be like analogous versus homologous.
Mhm.
Homologous, on the other hand, are structures that have a similar evolutionary history, but might now have different functions. So, whereas analogous evolved differently and sort of the function is the same, homologous is similar evolutionary history and now possibly branching out into different functions. And so, we're looking in the second component, we're looking for something that evolved similarly but might be serving different functions here. So, the wing of a bird and the leg of a bird. No, those probably, why would those don't have like similar evolutionary histories? They're not even necessarily the same like structure, so to speak.
Yep.
The arm of a human and the flipper of a walrus. All right, that sounds reasonable. These are both mammals. Even though they're serving different functions, they're the same like anatomical like location.
It doesn't serve a different function when I'm in the water. I'm swimming with my arms and they're swimming with their flippers.
All right. Well, if you have, all right, I suppose so. If you're a strong swimmer, I struggle in the water. No, not actually. All right. So the arm of a human and the flipper of a walrus seems like this could be what we're looking for. Seems like, they evolved similarly. They're both mammals, but they certainly look a little different. Might serve generally different functions, unless you're, I guess, I don't know, swimming in the ocean or something like that. The wing of a bird and the wing of a bat, just like we talked about, this is analogous. And then answer option D, the wing of a bee and the wing of a bat, that's also analogous. So for those reasons, we're eliminating C and D because that second answer, or the second portion of that answer, represents what an analogous structure is rather than homologous. So really, it's just between A and the second portion of A and B, trying to realize which one or which of these have similar evolutionary histories, but possibly now different functions. And so for that reason, I go with B because, even though they evolved separate, or they evolved to have different functions now, again, unless you're swimming, I guess. The arm of a human and the flipper of a walrus do have a very similar evolutionary history because they're both mammals.
Oh wait, so I'm confused. I got this one right, didn't I?
Did you pick B?
I think so. I don't know. I forget. Oh, maybe I picked C. I don't know. I like B though. Yeah. Maybe I picked C. Oh well, I picked C. C and move on as the story goes.
The old adage says, "See and move on."
Yes. Wow. All right. It's a brutal end to biochem. Trying to think about analogous versus homologous, like one random piece of knowledge that we learned at some point in biology or evolutionary whatever class that was in.
Yeah, it just goes to show they can really, especially on the discrete section, they can throw in a question that is just out of left field, right? Analogous and homologous structures. You got to be ready for everything.